So we can get a hybrid plant. That is called as somatic hybrid. Why it is called as somatic hybrid? Because we are using the somatic cells, it is called as somatic hybrid. The process is called as somatic hybridisation. So the process is called as somatic hybridisation. So it is having lots and lots of application to produce a hybrid plant. Okay, so you know hybrid plant means we can good quality from one plant and another plant. We can fuse it and we can get a high variety of the plant. So we get the somatic. So that is the application of this protoplast fusion. So first you know you are taking a strain A. The first one, strain A, a different character. For example, it is having a good color of the fruit. This particular plant, strain B, is having a good taste. So we can get the color and we can get the color and the taste of the fruit. Both we can get. Like for example, I said, so we are taking this strain A and strain B. First thing is strain A, strain B. We are taking. These are having good qualities. Okay. So the strain A, what we are going to do, we are going to isolate the protoplast. First is the isolation of the protoplast. First is isolation. Why we need to isolate? We can get the isolate. I can isolate the protoplast. First we are going to isolate protoplast from strain A. We can isolate protoplast from strain B. You know what are the techniques we are using for isolation because we already studied here. Okay. So we are going for enzymatic method and the mechanical method we are doing. So mechanical method, if we are doing physical, we are going to chop and cut it and we are going to centrifuge it. Okay. So that is a mechanical method. So we can go for enzymatic method because already I said enzymatic method is having a importance because it will, the cells are not getting damaged. So we are getting a good quality of the protoplast. So here isolation of the protoplast is the first method. Okay. First you are going to isolate. So now we are having the protoplast in our hands. You know method already I discussed over there. Okay. So now we are going for next fusion of the protoplast because we are having two protoplasts with us, A and B, strain A and strain B. For strain A, like that same peeling, we are taking the leaf. I said why we are taking the leaf because it's so easy. We are peeling the leaf, plasmolized cells will be getting. We are making into small pieces. Then we will put the enzymes over there. After that, we are using the osmolytic substances to maintain the osmosis of that particular cells. Because once the protoplast is formed, it will damage and it will come out. Okay, all the cell organelles will come out of the cell because now it's so sensitive. Okay, okay. So now we are having protoplast A and B. So now what we are going to do? We are going to fuse this protoplast. So this is the importance of this protoplast. So very easy. When it's having a cell wall, it's difficult to fuse two cells. That's why we are going for this protoplast. So when protoplast is so easy for them to combine together. That's why we are going for the protoplast isolation and fusion. Okay. So next step is first is, as I said, isolation. Next is fusion. So like one paragraph if you write this topic will get over. Okay. So coming to the fusion. So how we are going to fuse this one? So here two different fusion methods are there. One is called as spontaneous fusion. Spontaneous fusion. Another one is induced fusion. Spontaneous means on its own it is getting fused. When you keep this strain A and strain B together, it will fuse on its own. They will come near and they will get fused. Okay, it will get fused. No need to do anything. But it takes some time. But how much percentage it's getting fused, we don't know. We are not doing anything. Just we are putting both strains, protoplast together and we are just leaving it. Okay, under this osmolytic substances, DMSO and sucrose because it should not burst. So when they are putting, automatically it will fuse. We don't know how much it is fused, how long, because we have done so much of steps to isolate the protoplast, right? Then we don't want to waste that time. Then they can go for induced fusion. Okay, so induced fusion reaction means all of you know we are going to give some external substance, okay, which will fuse it together. Okay, that is what is induced fusion. This induced fusion can be done by 3 different methods. Okay, one is they are using sodium nitrate. Second, they are using calcium ions. Third, they can use electricity. Can you follow, girls? Are you following?
Yes, ma'am.
Girls, the online class, are you following? Alright. So first is sodium nitrate. They will be using sodium nitrate and they will be using calcium ions and they will be using electricity. Less amount of electricity. Okay. And they will be using a fusogenic agent called as PEG. This is a 1-mark question. Polyethylene glycol. PEG. So induced fusion. Now we are having this density gradient certification is that the protoplast will be present over there. They both are fixing here and they are fusing it and they are using sodium nitrate, calcium ions or electricity. Okay. Also that what happens is both will be inducing. Okay. It will come together and it will start fusing together. They will get fused. Okay. And you will get a zygote. One cell you will get. Okay. So you will be using polyethylene glycol. PEG. After fusing, what we are going to do is we are going to verify this method. That's what. So we verify whether it is getting fused or not. Okay. So for that we are using some kind of test they will be doing whether it's getting fused. They should see under the microscope. Yes. And they will be using some dyes and all they will be using. And they will be taking remove and they will see under the microscope whether it is getting fused or what. Okay. So here what they are doing. Then we are going to see this is a cell which is a single cell. This is pink color and yellow color. 2 different cells are there. So here it will get fused and first it will become single cell. It is there. Then fusing. 2 are getting fused. How much getting fused? It can be sodium nitrate, calcium, and they use PEG, that is polyethylene glycol, and after that they can use electricity. This is an option. But first they will be using the chemical. You can tell it is again chemical and the physical process also. So physical process, low amount of electricity. So this low amount of electricity, what it will do? It will weaken the cell membrane, okay, or the plasma membrane of the particular protoplast. So when it's weakened, what happens? These both can come and attract each other and they can fuse also. Okay, so like that they will be fusing, then single, double, and after that it got fused and it you can see here 2 protoplasts are fused. One is having the pronuclei yellow color and another one is the pink color. So it's fused. Once it is, this takes nearly only 2 days to get this one. Within 2 days they get within 2 days or hours also sometimes it may get. So you can see outside cell wall slowly it is forming. Bit and bit. Together it will not form. Okay, so after that, till they will be using the sucrose and DMSO to maintain the stability. Okay, and after that you will get a fused protoplast. Now, after this fused one, this will be having both the nuclei. It's having both the nuclei. That you should remember here. Both the nuclei. That's why it is called as hybrid. This we are getting from a somatic cell. It is called as somatic hybrid. The process is called as somatic hybridisation. Understood? Somatic hybrids. When it is hybrid, when both the nuclei are, the pronuclei are formed together, see this is one nuclei, this is one nuclei. This is for one cell, this is for another cell. So now we are having 2 nuclei in that together, fused. Okay? So it is called as, then after this fused one, it produces a first division. Okay? The cell, it will become 2 cells. Then after that the clumps of cells are formed and after that you are getting full cells, then you will get a callus tissue. So this callus tissue, what they will do, they will identify, each step they should identify, not only at the last step, because we are doing so much research over here. Okay, here they should see whether it is fused or not. Same cell should not fuse right here. From strain A only it should not fuse. Strain B only together it should not fuse. Strain A and strain B together it should get fused. Are you following? So identification is important. Then after that here also, once it is done, Whether the fused cells are only getting the callus of what clumps of cells are formed, that also you should identify. Because that cells only will give rise to a hybrid plant, somatic hybrid plant. Are you following? So like that the callus is formed. After that callus, you know, direct regeneration and indirect regeneration. After callus, you know, you can do hundreds and hundreds of work over there. Okay, so next after that the callus is being differentiated after the callus tissue. It's directly you get the shoots on that, or sometimes we can go for the embryos above that. That embryos we can go for the embryogenesis, somatic embryogenesis, that and only. But basically here after this callus you get the shoot like that. The shoot we can put it in the shooting media. Then after the shooting media, MS media, you know what are the steps we should follow? We should follow the in vitro culture steps, all like maintaining the temperature, maintaining the pH of the media, maintaining the light, everything you should maintain, okay? All this will be maintaining and you get a regenerated plant. This regenerated plant will be a somatic hybrid plant, which will have the quality of A and B strain. So as I said, this is having some fruit, for example, I said red colored, nice color. Another strain is having the same plant, okay, the same plant only we are going to. It's having the taste. So now we can get the color and the taste of the plant together in a hybrid. This process is called as somatic hybrid, and the process is called as somatic hybridisation. So this is an excellent example of the protoplast. So for going for the protoplast fusion, we should go for the isolation step process, which I discussed earlier. Then we will get this particular plant. Very, very important 20-mark question, big long answer. And so now what we can see, both the nucleus are fused. That's why it is called as hybrid. Suppose one nucleus we can remove. Only one nucleus of one plant is there. Other cytoplasms are fused. From this plant, this plant. First, suppose strain A ka nucleus they are fused. Strain B nucleus they removed. Are you following, girls? Other cytoplasm inside the cell, whatever is there, that is there only. Mitochondria, chloroplast, everything is there. Okay, ribosomes, everything is there for both the plants. But only one plant, if they remove the nucleus, this together it is called as hybrid. Cytoplasmic hybrid is called as hybrid. Cytoplasmic hybrid. Okay, this also have certain application. Why? Because both the nucleus sometimes it may not give that much good effect. So which is a dominant, very dominant plant, or which is having a good this thing, they will select that particular nucleus of that plant, and another nucleus they will remove that one. Okay, in the first one they remove the nucleus such that it will not have nucleus. One plant will not have, one strain will not have nucleus, another strain will be having the nucleus. So these are called as hybrids. So cytoplasmic hybrids are called as hybrids. So same after that hybrids, they will get all these plants. So that plant will be a hybrid plant. It will be having lots and lots of application in that. Okay, for studies about the chromosome. Okay, like cellular level, how it's developing, like that. So many research are there for hybrids. So they are going for the hybrid plants. Right. So this is all about this somatic hybridization. Yes. Any doubts there, girls? Any doubts, girls, in the online class? People can ask me.